Hello everyone and welcome back to my channel. So today we've got our final GCSE protein synthesis video, and today's video is for OCR GCSE specifications. So that's for Biology A and B Foundation and Higher, and also Combined Sciences A and B. So let's get into the video. Our first set of specification points are for OCR GCSE Combined Science A and Biology A. And these are number 1, inside every cell is genetic material, and this is used as a code to make proteins. And number 2, explain the importance of amino acids in the synthesis and breakdown of proteins, use of the terms monomer and polymer. For our first point, as you can see here, we have our cell, and within the nucleus of the cell is genetic material known as DNA. And a section of DNA is known as a gene. And this gene contains a code, and it is this code that determines the amino acid sequence which influences the specific protein produced. And for our second specification point, here we have our amino acids, and the amino acids are monomers, and monomers join together in order to form polymers. So we've got our amino acids, and protein synthesis occurs, which leads to the formation of new proteins. So these amino acids are joined together, monomers are joined together, and polymers, which are new proteins, are produced. So now we're going to move on to some questions based on that information. So question 1, what are proteins made of? And then we've got some answers there. So we know that proteins are polymers and they're made up of monomers, and these monomers are amino acids. So it's amino acids that join together in order to form proteins. So therefore our answer is amino acids. Okay, so now we're going to move on to the Biology A Higher further specification points. Number 1 is to recall a simple description of protein synthesis, the unzipping of the DNA molecule around the gene, copying to mRNA in nucleus, transcription, translation of the nucleotide sequence in the cytoplasm. And number 2, to explain simply how the structure of DNA affects the proteins made in protein synthesis, triplet code, and it's used to determine amino acid order in a protein. And the Biology B higher tier further specification points are number 1, to explain simply how the sequence of bases in DNA code to the proteins made in protein synthesis, including the idea that each set of 3 nucleotides is the code for an amino acid. And number 2, recall a simple description of protein synthesis in which a copy of a gene is made from messenger RNA, mRNA. The mRNA travels to a ribosome in the cytoplasm. The ribosome joins amino acids together in an order determined by mRNA. And learners are not expected to recall details of transcription and translation for this one. Here we have our DNA molecule, and we've got a section highlighted which is known as a gene. A section of DNA is a gene. And a gene is made up of bases, and these bases are A, T, G and C within DNA. And the code that determines the amino acid sequence is known as the triplet code. So what this means is that for every 3 base sequence, a specific amino acid is coded for. So for example, we've got TAC here, and that codes for the first amino acid. CGA codes for another amino acid, TGC for another, and so on. So it is this base order which controls the order of amino acid assembly. So it controls the order amino acids are linked together. So we've got our bases here and the specific amino acids they code for. And so it's the amino acid sequence which then determines the protein produced. Okay, so now we're going to talk a little bit about the actual process of protein synthesis. So the first stage of protein synthesis is transcription, and this begins with our DNA molecule, and we've got our gene selected there, and the DNA actually unwinds or unzips around the gene, and the DNA strands separate at this point. Now, the genetic material within eukaryotic cells is stored within the nucleus, so this is where transcription takes place. Now, after the DNA strands have separated, the second DNA strand, which is the template of the gene code, is actually copied into mRNA. Now, this occurs by the formation of hydrogen bonds between bases. So on the DNA strands, there are A, T, C and G bases, whereas on the RNA strand, there are U, A, G and C bases. So A on DNA complementary base pairs with U, T complementary base pairs with A, C with G, and G with C. Once the mRNA strand is formed, it actually moves out of the nucleus into the cytoplasm, ready for the next stage of protein synthesis, which is translation. So translation begins with the mRNA travelling to a ribosome in the cytoplasm. And it is at this ribosome where translation of the nucleotide sequence on the mRNA molecule occurs. And the ribosome joins the amino acids together in an order determined by the mRNA code. So now some questions based on that information. Question 1. During protein synthesis, the DNA that codes for a particular protein is copied. This copy is called mRNA. The diagram below shows the base sequence for a section of DNA. What would be the complementary sequence of mRNA for this section of DNA? So we know that DNA is made up of the bases C, G, A and T, whereas mRNA and RNA itself is made up of C, G, A and U, not T. So if we have a look at our possible answers, we can see that A and C contain T bases. So we know that the mRNA structure mustn't be A or C because they contain T bases. So we can discount those. Okay, so if we have a look at the sequence itself, so we've got G, G, T to begin with. So if we have a look at D, we see we've got G, G, U, and we know that G complementary pairs with C and T complementary pairs with A. So we know that D cannot be the correct corresponding mRNA sequence because these are not the correct complementary bases, so we can discount D. Now let's just check that B is our correct answer. So we've got G, G, T, and then we've got C, C, A. So we know that G complementary pairs with C and T with A, so that is correct. Then we've got G, C, and A, and these do complementary pair with C, G, and U. And then finally we've got T, A, and T, and these do complementary pair with A, U, and A, which means that our answer is B. Insulin is a protein made up of 51 amino acids. How many bases are in the length of DNA coding for insulin? So we know that 3 bases code for 1 amino acid. So this means that if we've got 51 amino acids, we've got 3 times the amount of bases. So if we do 51 times by 3, we get an answer of 153. Question 3. Switching off the protein mechanism described in Part B involves the plant making a protein. Now I know that Part B isn't included on this slide, but if you head over to my website you will find that extra piece of information in the question pack. However, you don't need to know what Part B says in order to answer this question. So scientists have put extra copies of the gene for this protein into the plants. This makes the plant make more mRNA molecules. Explain why making more mRNA will switch off the mechanism faster. Now this question is more about applying your knowledge of protein synthesis, so it may seem a little bit confusing at first, but if we just go through and highlight the key points, then this should make the question easier to understand. So it says that switching off a mechanism involves the plant making a protein. So these scientists have put extra copies of the gene for this protein into the plant, and this is making the plant make more mRNA. And then it says, explain why making more mRNA will switch off the mechanism faster. So we know that mRNA is a copy of the gene code, and it is translated in order to make our protein. So therefore, our answer is going to be that mRNA carries the code for proteins, and therefore more protein will be made. Question 4. Amaya reads an article in a magazine which explains that genes code for the production of a taste receptor on the tongue. Taste receptors are proteins. Complete the sentences to describe how a protein is made. And then we've got a selection of keywords there. So let's have a look at the first sentence. So a copy of the blank is made from blank. So we know that the first stage of protein synthesis is transcription, and initially the first stage of transcription is that mRNA is copied, and this is copied from a gene, which is a section of DNA. So therefore our first sentence answer is going to be a copy of the gene is made from mRNA. Then our second sentence, so this molecule, so therefore it's going to be this molecule referring to mRNA, travels to a something in the cytoplasm. So we know that when translation first begins, the second stage of protein synthesis, mRNA actually travels to a ribosome in the cytoplasm, and this is the site of protein synthesis. So therefore, our answer is going to be ribosome. So the next sentence says, here, blank, are joined together to form a protein. So we know that it is in fact amino acids that are joined together to form a protein. And then finally, there's a sentence about mutations, which I haven't covered in this specific PowerPoint content. However, just for the sake of completing the question, we're going to have a little read through. So a mutation would create a something and therefore a different receptor. So a mutation would cause a change in the amino acid sequence due to a change in the DNA code, and therefore a different protein is going to be produced. So the answer is going to be genetic variant, as a mutation would create a genetic variant and therefore a different receptor. And here we can see our answers. Question 5. RNA is another type of nucleic acid. It is involved in protein synthesis. The table shows the nucleotide sequence in RNA that codes for different amino acids in proteins. So then we've got a column of amino acids, we've got their abbreviations, and then we've got their nucleotide sequences. And then it says, look at the nucleotide sequence in a section of RNA. Write down the sequence of amino acids that this section of RNA codes for. Use the abbreviation for each amino acid. So again, this question has a lot of information in it, so it may seem a little bit confusing at first, but it's actually quite simple. So firstly, if we just have a look at our nucleotide sequence, so we've got a range of bases here. Now we need to find the bases that code, that match up, to these nucleotide sequences here. So if we start and we go across, so we've got GAC, so GAC is not included here, so then we'll go along to the next base. So we've got ACU, which is also not included here. Then we've got CUG, and CUG is here, and that is in fact leucine. So we know that our sequence actually begins here at CUG. So our first answer is going to be LEU, because remember we're using the abbreviation for each amino acid. So Lu is going to be our first answer. Then we've got GAG, which is here. So that is coding for glutamic acid, which is glue. So our second answer is going to go here, and that is going to be GLU. Then our third amino acid, so we've got UAC, and UAC is here, and that is tyrosine. So therefore our answer is going to be TYR for tyrosine, and that will go here. And then finally we've got ACG, and ACG codes for threonine, and that is THR, so therefore that will go there. So you can see our answer here. Question 6. Antibodies are proteins. The statements below describe protein synthesis. We've then got 4 statements, and the question is put the statements in the correct order. So if we have a look at the statements, we've got A, which a copy of the gene is made from messenger RNA. So we know that that is a stage of transcription, so that is likely to be at one of the first statements in the order. We've then got B, which is the gene that codes the protein is found in the DNA. And we know that's actually the first stage of transcription, so it's very likely that B is going to be our first answer. But let's just check through C and D to check. So C says that mRNA travels to a ribosome in the cytoplasm. So we know that this is the first stage of translation, so this occurs after transcription. And then D, the ribosome joins the amino acids together in the correct order. So we know that's the final stage of protein synthesis and the final stage of translation. So we know A and B are going to be our first 2 statements, and then C and D are going to be our 2 final ones. So we just need to work out the correct order. So let's have a look at B. So B is the gene that codes the protein is found in the DNA. So we know that that is the first stage of protein synthesis and the first part of transcription, so that's going to go here. Then we've got A, so a copy of the gene is made from messenger RNA, so this occurs second in transcription. And then we move on to translation, where the mRNA travels to a ribosome in the cytoplasm, so we know that comes next, so C is here. And then finally D, the final stage of translation and protein synthesis, is the ribosome joining the amino acids together in the correct order, so D is going to come here. So you can see our answer there. So B, A, C, D. Now, if you do have a look at this question on the actual sample paper, there's actually a mistake in the mark scheme, so this is the correct order of statements. Question 7. DNA structure is important in the production of proteins. DNA analysis allows scientists to group organisms based on similarities in their DNA. Complete the sentences below. So our first sentence is a blank of bases is required to code for an amino acid. So we know that the triplet code is a structure of DNA. So it is in fact a triplet of bases required to code for an amino acid. So a sequence of 3 bases codes for an amino acid, so therefore our answer is a triplet of bases. And then the second sentence, the properties of the protein may depend on the blank of the amino acids. So we know that it's in fact the sequence of amino acids which determines the specific protein produced, and therefore the properties of the protein. So therefore our answer is sequence of amino acids. And then finally our last sentence is not linked specifically to the content that I've been talking about, but I will be doing a video on DNA and RNA soon, so this is probably more relevant for that, but just to complete the question we will go through it. So grouping organisms according to similarities in their DNA or physical characteristics is called blank. So if we're grouping anything, really this word or term can be used, so the answer is in fact classification. So when we're grouping different organisms or grouping anything, really it is in fact the classification which determines these groups. And there we can see our answers there. So now we're going to move on to the OCR GCSE Combined Science B and Biology B specification points. So there are quite a few here, so I'm just going to go through them quickly. So number 1, genes tell a cell how to make proteins by joining together amino acids in a particular order. Number 2, explain the importance of amino acids in the synthesis of proteins, including the genome as instructions for the polymerization of amino acids to make proteins, use of the terms monomer and polymer. Producers make glucose using photosynthesis. Some of the glucose is used to make proteins for growth. Number 4, photosynthetic organisms take in nitrogen to make proteins. Number 5, producers take in nitrogen compounds from their environment and use them to make small organic molecules, including amino acids. These small molecules are used to make larger organic molecules, such as long-chain proteins. Explain the importance of amino acids in the synthesis and breakdown of proteins. So for our first point, here we have our DNA molecule, and of course a section of DNA is known as a gene, and it is in fact the genes that contain a code which tells a cell how to make proteins, and this is by determining the amino acid sequence. So the code influences the amino acid sequence, which then determines the specific protein produced. So for our second point, here we've got our amino acids, and these are monomers, and protein synthesis takes place and new proteins, which are polymers, are created. Now it's important to note that the genome, which is the entire genetic material of an organism, actually is the instructions for the polymerization of amino acids, which then make proteins. So the genome contains the genetic code, which tells the cell the amino acid sequence, which then determines the protein produced. So producers and photosynthetic organisms, so plants for example, need to obtain nitrogen and glucose in order to produce amino acids. Amino acids are therefore important for protein synthesis, so without nitrogen and glucose, amino acids wouldn't be able to be made and protein synthesis wouldn't be able to occur. So producers slash photosynthetic organisms, they take in nitrogen in the form of nitrate ions, and this is taken up from the soil through their roots. Then glucose is made through photosynthesis, and so glucose and nitrate ions are then used in order to make proteins, which are important for growth and also tissue slash cell repair. So as you can see here, we've got our nitrate ions plus our glucose equals our amino acids. So here we've got our producer taking up nitrate ions from the soil, and these nitrate ions are used to make small organic molecules such as amino acids. These amino acids, which are small organic molecules, are then used to make larger organic molecules such as long-chain proteins. So amino acids are very important in protein synthesis and also in protein breakdown. So amino acids are linked together by peptide bonds in order to form new proteins, and then when the bonds are broken between these amino acids in the new proteins, then there is a protein breakdown into amino acids. So now we're going to move on to some questions based on that information. So question 1, what is a genome? And then we've got a selection of answers there. So we've got A, a description of the number of chromosomes in an organism. B, all the proteins that one organism can produce. C, a store of seeds to preserve genetic variation. Or D, the entire genetic material of an organism. So if you remember back, we remember saying that the genome contained the instructions for the amino acid sequence. So the genome is actually the entire genetic material of an organism. Question 2. Retinitis pigmentosa is a genetic condition that affects the eyes. It is caused by a mutation to a gene. The mutation produces a recessive allele. The condition causes rod cells in the retina to break down. Explain the meaning of these terms. Gene. So we know that gene is in fact a section of DNA or a length of DNA that codes for a specific protein. So here we have our answer there. So a length of DNA that codes for a protein. Question 3. Scientists use key terms to explain genetics. Use words from the list to complete the sentences about genetics. So we've got some keywords there. So then this passage is the molecule which carries the genetic information is DNA. DNA is a polymer made up of blank. So although we haven't covered what DNA is made of, I will be doing a video on DNA and RNA. So just for the purpose of this question, the answer to this first one is nucleotides. So DNA is made up of nucleotides. Nucleotide is made up of a phosphate group, a pentose sugar, and also a nitrogenous base. So our base is A, T, G, and C. So that is what one component of the nucleotides in DNA is. Then we've got sections of DNA which code for a particular protein are called blank. So we know that sections of DNA which code for particular proteins are, of course, genes. So there we've got our answer. Question 4. So this is a very similar question to the previous one. This one is just included on the higher tier paper. So it includes an extra sentence, and there's actually more leeway with the answers you can give. So if we go through, so DNA is a polymer made up of, so previously we said nucleotides. On this question, nitrogenous bases, as I mentioned before, is also accepted. And then sections of DNA which code for a particular protein are called. So also on this question, you can put codons and something else, which I can't remember. We'll have a look at the answer in a second. And then the last sentence is an organism's entire genetic material is called its something. So we know that the entire genetic material which provides the instructions for the amino acid sequence is known as the genome. So there we go. Yeah, so for the second one, sections of DNA which code for a particular protein are called, you can have genes, codons, or base triplets as well. Question 5. Nina is learning about substances absorbed by plants. She finds out that plants absorb nitrate ions from the soil. Explain why nitrate ions are essential for plant growth and survival. So we know that nitrogen is an essential component of amino acids, which are important to make proteins. So nitrate ions absorbed from the soil is the plant's only source of nitrogen, and therefore its only way that it can obtain nitrogen in order to make amino acids, which affects the proteins produced. So here we can see our answer. So nitrate ions are the plant's only source of nitrogen to make amino acids / proteins / nitrogenous compounds. Question 6. Alligators eat fish, birds, turtles and snakes. These foods are high in protein. Put a tick in the box that describes what proteins are made of. So we know that proteins are polymers and they are made up of monomers, which are in fact amino acids. So therefore our answer is amino acids. So thank you so much for watching. Please subscribe to my channel. It means so much to me. If you could like this video, share and also comment down below any other videos you would like me to film, please do. Also head over to my Instagram and give me a follow to get updates of when I've posted on my website and also on my YouTube channel. And most importantly, head over to my website where you can find lots more exams questions and revision resources for OCR, AQA and Edexcel, GCSE and A-level. So thank you so much for watching guys, and I'll see you in my next video. Bye!